Hello, today I will be explaining the muscle histology, specifically of the skeletal muscle. So let's begin by explaining the kinds of muscles that are three kinds of muscles that are in the human body. The first is the skeletal muscles, and its fibers are striated, tubular, and multinucleated. It is voluntary and usually attached to a skeleton. The second type of muscle is a smooth muscle, and its fibers are non-striated, spindle-shaped, and uninucleated, which means that they only have one nucleus. They're involuntary, which means that they cannot be controlled consciously, and they usually cover a wall of internal organs. The third type of muscle is the cardiac muscle, and its fibers are striated, branched, and uninucleated, just like the smooth muscle, and it's also involuntary, just like the smooth muscle, and it only covers the walls of the heart. And here's an overview of skeletal muscle cells. A myocyte is a muscle cell. A sarcolemma is the cell membrane of a muscle cell. The sarcoplasm is the cytoplasm of a muscle cell containing organelles and myofibrils. The sarcoplasmic reticulum is the specialized endoplasmic reticulum that stores calcium ions. A myofibril is a long thread-like structure that runs parallel to the muscle fiber and contain myofilaments. And myofilaments consist of two types of filaments. The first is a thick filament, and which is composed mainly of myosin, and thin filaments, which are composed mostly of actin, along with troponin and tropomyosin. And these are all proteins. Myosin, actin, troponin, and tropomyosin are all protein. And this picture shows basically what we will explain later, which is their sarcomere. The striated appearance of skeletal muscle fibers results from the organized arrangement of actin and myosin myofilaments in a sequential pattern that extends from one end of the muscle fiber to the other. This highly organized structure of proteins forms a sarcomere, which is the functional unit of a skeletal muscle cell. And each sarcomere is contained with a myofibril that spans the entire length of the muscle fiber, and it connects to the sarcolemma at its ends. So myofibrils are approximately 1.2 micrometers in diameter, and this allows them to have hundreds of them or hundreds of thousands of them within a single muscle fiber, containing thousands of sarcomeres. Each sarcomere measures about 2 micrometers in length, and they have all a cylindrical three-dimensional structure bordered by Z discs, which are also called Z lines if the diagram is represented in two dimensions, like this one, to which the actin myofilaments are anchored. The actin and the troponin tropomyosin complex, which extends from the Z disc towards the center of the sarcomere, form strands that are thinner than the myosin. So, and this is why they're called a thin filament. And on the other hand, the myosin strands have multiple heads projected from the center of the sarcomere, but not reaching towards the Z disc, and are thicker and are more massive, earning them the designation of the thick filament. And we will describe how these sarcomeres are involved in movement later on in the slide called the sliding theory of movement. T-tubules are crucial in the functioning of a muscle cell. They are extensions of the sarcolemma that penetrate into the muscle cell, and they facilitate the transmission of action potentials. As you can see here, the T-tubule is filled with extracellular fluid, and they allow the sarcoplasmic reticulum to reach into the individual's uh, sarcomeres. And as you can see, the sarcoplasmic reticulum and the T-tubule form a structure called a triad. And when they're together, and the T-tubule helps the sarcoplasmic reticulum to to get to the sarcomeres more easily, and they're crucial for muscle contraction, as you can see here. For a muscle cell to contract, the sarcomere must shorten in response to signal from the central nervous system. While the thick and thin filaments do not actually shorten, they actually slide against one another, leading to the shortening of the sarcomere while the filaments maintain their length. And this is called the sliding filament model of muscle contraction. The contraction mechanism involves the binding of myosin to actin, which forms so-called cross bridges to that facilitate filament movement. And these cross bridges, which are the connections between actin and myosin, enable the myosin heads to pull the thin filaments closer to the end line, which shortens the sarcomere. So, as the sarcomere shortens, some areas contract while others remain unchanged. And a sarcomere is also defined as a distance between two adjacent Z disks or Z lines. And during the contraction of a muscle, the distance between the Z disks decreases. And the H zone, which is located in the center of the A band and only contains thick filaments, also shortens during contraction. So in a fully contracted sarcomere, the H zone may completely vanish as the thin filaments are drawn all the way towards the hemline, and the I bands, which are only thin filaments, so they only contain they don't have any myosin. They shorten the overlap between the thick and thin filaments. The A band really actually does not shorten at all. It is the band that actually doesn't change length at all because it has thick filaments which do not slide at all. However, s- the A bands of the adjacent sarcomeres move closer together during contraction. The thick filaments pull the thin filaments towards the center of the sarcomere until the Z discs approach the thick filaments. The zone of overlap where the thin and thick filaments coexist expands as the thin filaments move forward. This process is also energy intensive, requiring the use of ATP, as you can see in this diagram here. The myosin heads split ATP and become reoriented and re-energized, and this allows the myosin heads to bind to actin, forming cross bridges, as we mentioned earlier. The myosin heads rotate towards the center of the sarcomere, which is called a power stroke also as well. The power stroke then pulls the thin filaments towards the center of the sarcomere. As myosin heads bind ATP, the cross bridges detach from actin, and again the myosin heads split ATP and become reoriented and energized, and this cycle just goes on again. And this complicated process happens every time we move a muscle in our body. So the changes during contraction versus relaxation are as follows. So during contraction, the sarcomere shortens significantly, and the I band and the H zone decrease in size. While, as I mentioned earlier, the A band remains the same because it is formed mostly of thick filaments which do not slide at all. During relaxation, the sarcomere returns to its original length, and calcium ions are pumped back into the sarcoplasmic reticulum. We'll explain the role of calcium ions within muscle cells later in this presentation. So, ions are crucial in muscle contraction. They're released from the sarcoplasmic reticulum during action potential, and they bind to troponin, which causes a conformational change that moves tropomyosin away from actin binding sites. So, um, within an actin filament, the tropomyosin is the basically covers the myosin binding site of actin, and the troponin controls the tropomyosin, and the calcium ions control the troponin. So it is cascading levels of control, and the calcium ion is at the top of it. The calcium ions are at the top of the hierarchy of this control, and this allows the actin filaments to bind to the myosin of the thick filaments. Overall, the key takeaways are understanding the structure of skeletal muscle cells is essential for grasping how muscles contract. The sarcomere is a fundamental unit of contraction, and its components play specific roles in muscle function. And the sliding filament theory provides us clear explanation of the contraction mechanism. Here are my citations. I used the OpenStax college textbook that was provided in the class. Thank you. Thank you very much for watching this presentation.